Good morning, students. So this is the second part of your chapter number 9, strategies for enhancement in food production. Okay, so today we are going to start with types of hybridization. So types of hybridization mainly of three types. First is intraspecific, second interspecific, and last is intrageneric hybridization. Okay, so I will tell you what is interspecific hybridization. Okay, so in very short way, you just understand that subspecies will cross with the species. Okay, so this is called intraspecific hybridization. Okay, so the cross between two varieties or subspecies of same species, means the cross between the same species or with subspecies. This is known as intraspecific cross. Okay, so for this example, intraspecific cross, so many varieties of rice paddies has been developed by crossing two subspecies of rice. Okay, so this is basically known as intraspecific hybridization. So this is your first type. Second is your interspecific hybridization. So what is interspecific hybridization? When the cross is done with the offspring. Okay, so two different species of same genus. Okay, two different species of same genus is called the interspecific hybridization. Is it clear? So this is known as interspecific hybridization. So here we have one example of interspecific hybridization. That is, see here in this example, this is what? This is Brassica oleracea and Brassica nigra. Okay, so these are two parents. Okay, these are two parents, but so you can see the value is this Brassica oleracea is cc is to 2n is equals to 18, है ना? And this is crossed with nigra. Okay, that is bb is to 2n is equals to 16. Okay, when they will cross with each other, so our first generation, that will be hybrid. Okay, so it will form 1b from here is from 1c and here is from 1b. So this is called b and c is to n is equals to, so what is common number is common number 16, then after this 17 and this 18. Okay, so here they form the mixture of the two parents. Okay, so but the thing is, this is hybrid in the first generation, but this is sterile. Sterile means not able to produce or not able to do the reproduction. Okay, so what we will do, we have already seen this technique, that is cloaking technique. Okay, so in this treatment we will provide this treatment. So what we will get, the new species we will get, and that name is Brassica carnata. Okay, so this is called fertile. So what is the result of this fertile or new species? That is bbcc is to 2n is equals to 34. When we will add 18 plus 16, so our answer will be 34. Okay, so this is known as interspecific hybridization. Okay, so next is your intrageneric hybridization. So what is intrageneric hybridization? When plant and two different genera of same family, got it? Plants crossed with two different genera of same family. Okay, this is known as intrageneric. So crossing between two different genera of the same family is referred to as intrageneric hybridization. Okay, so it also having many types of important intrageneric cross. There are types of important intrageneric cross. So they are, first is radish cabbage cross, and second is our wheat rye cross. Okay, so we'll start with the first point, that is radish cabbage. Okay, so here we will understand by this example. See here, this is for in our one side we will take radish, and one on the other hand we will take the cabbage. Okay, so when these two will crossed with each other, the result will be hybrid and sterile, means again they can't able to reproduce, okay, unable to reproduce. So again we will give the treatment, right? So that treatment will give us a result that is known as amnio-diploid, okay. So this will be our result, okay. So this is the example of radish and cabbage. But so here we having another example, that is wheat and rye cross, okay. So in this, again look at in this example. So what is this? Is Triticum aestivum, and this is Sickle cereals, okay. So in this example again we having the cross, okay, the cross between two genera of the same family. So again we will get the result, है ना, that will also not same, okay. What we actually want, it is not like that, means even our first generation again it will form a sterile hybrid. So the result is 2n is equals to 28, okay. So again we will give the treatment, and from our F2 generation, means whatever the F2 plant will be formed, that is formed, that this is also known as amphio-diploid, okay, amphio-diploid. You can see here 2n is equals to 56. When you will add 42 with 14, you will get the result 56, okay. So means after the treatment we can find the exact result what we want, okay. So these are the basically three types of hybridization. So here in this table you can see different between intraspecific and intrageneric hybridization, okay. Now the significance of intergeneric species and intergeneric cross, okay. So what are the basic significance? So there are two useful example of intergeneric hybridization. The first is tritical and another example is maize tripsicum cross, okay. We have already seen this example, right? Okay. So now come to the fifth point, that is known as mutation breeding. So what is mutation breeding? So mutation breeding is a sudden heritable change in a characteristics of an organism, means we can see the change in the characteristics of any organism, such as a change may occur due to change in genes, or maybe can say in chromosomes, okay. So mutation may be of spontaneously or induced. So the utilization of induced mutation for crop improvement is known as mutation breeding, means when we will do the improvement of that crop is known as the mutation breeding, okay. So there are also many types of example we having, you can see here. These are the examples, okay. So next is our polyploidy breeding. So what is polyploidy? Okay, so if an organism contain more than two sets of chromosome is called polyploidy. Remember this, when we having more than two sets of chromosome, okay, and the situation is known as polyploidy, okay. So on the basis of number of chromosome sets, polyploidy may be on the basis of the number, okay. They may be 3, so 3 is known as tetrapolyploidy, maybe 4, that is known as pentapolyploidy, 5th that is hexapolyploidy, है ना. Or if so in this way, okay, means tripolyploidy, tetrapolyploidy, penta and hexa, okay. So it may be of two kind, okay, that is autopolyploidy and allopolyploidy. So what is auto and what is allo? Autopolyploidy, in this type of polyploidy, the extra chromosome sets belong to the same species is known as autopolyploidy, okay, means the chromosome will be of same species. It is produced due to irregularity in meiosis during gametogenesis or in spermatogenesis. So it is artificially produced by chloroquine, okay, the treatment which we have given, okay. So what are the significance of autopolyploidy? So autopolyploidy are very much useful for us. For example, seedless banana and grapes are autopolyploidy, and second is large size of watermelon and are autopolyploidy in nature, okay. So this is very important. Now next is our allopolyploidy. So what is allopolyploidy? Here the extra chromosome sent in a plant species come from another species. It is produced by cross between two species or genera. Tripolyploidy or allopolyploidy plant may be represent as, like see here in the example, maybe they can represent by AAB, है ना, when two genomes AA. Okay, so if they form one species or genome, then they will form B, means single. If they are one, then they will form B. If they are two genomes, then they will form AA, and if they are tri, then they will form AAB. Okay, so in this way, likewise. So the significance of allopolyploidy. So from evaluation and origin of new plant varieties and species point of view, allopolyploidy is very much important. It caused origin of many plant species and varieties. It can be understood by the following two examples. So first is your origin of brassica species, and second is origin of modern breed wheat. Okay, so we'll start with the first example. Okay, so origin of brassica species. Okay, so by the example you can easily understood what I'm saying. 3 diploid of brassica, okay, okay, so commonly found in nature. So their basic chromosome number are 10, 8, 9 respectively. Their genomes are represented by AA, BB and CC. You can see here, okay, respectively. A natural cross occurred between B, okay. So Campus test that AA and Nigra BB, through which the Brassica Jonesia AABB was produced. Okay, when AABB will cross, then they will form AA and BB, which was produced. Okay, so a similar cross also occurred between B Campisteria, that is AA, and B Ollerica CC. So given rise to the Brassica napus, that is AACC. Okay, so likewise in this way they will produce the next species. Okay, so another example we having the origin of modern bread wheat. Okay, so what we will see here, you can see we have wheat. In wheat is naturally allopolyploidy. It has two genomes, each T monococcums AA and BB, and also a squared rows, that is DD. Okay, so thousands of the year ago, a number crossed over between T monococcus, that is AA, to n is equals to 40, and the goat grass Aleolopes steroids, that is BB, 2n is equals to 14, and the sterile diploid hybrid, that is AB, was formed. So in this way you can see, here is also the example given. You can see by this example the cross and the result. So at last they will form the Triticum aestivum, AA, BB and DD, the modern breed wheat. Okay. Okay, so next topic is improvement in the species of wheat. How we will improve the species of wheat? In ancient time, 10,000 to 15,000 BC, only one species of wheat, that is Triticum, was present, right? But Triticum monococcums is another species of wheat which is used by man. Yes, both of these species were diploid and having only 14 chromosomes. So accidentally or suddenly, Triticum monococcus was crossed with a goat grass, and this cross is also diploid, that is 2n is equals to 14 chromosomes. So the offspring of T monococcus and P spiletoids is sterile. A polyploidy species having 2n is equals to 28 is formed from the sterile hybrid. Okay, so we having some improved varieties in the wheat. You can see here, these are the improved varieties of wheat. Okay, so in like this way we have improved the the species of maize also, है ना. So the present day maize plant produce grain locked in their cobs. Cobs means woody part of the maize, okay. Cobs with the further cover tightly by spreads. Spreads means sheathing bract of the flower, okay. So such an arrangement of great advantages of the farmer to the farmer. However, such an arrangement without the benefit of the seed dispersal disadvantages to the wildlife. The wild ancestors of the maize must have had the crossed cob to scale and to mature grain. It must have been very difficult for the early cultivators to pick these grains one by hand. So therefore maturations that cover the cob tightly by the spatches must have been spatches that bract in the flower, okay, must have been occurred in the past and must have been picked up by early cultivation to develop and improve varieties of the maize, okay. So here are the names of improved varieties in maize, okay. Now this is the second part, that is known as plant selection. How we will select plant? So I already told you how, है ना. So this is the method of selecting healthy and superior seed or fruit which are used for showering in the next year. It is practiced by all the farmers. They used to select healthy seed every year and use them as a propagule for the next crop, okay. So there are many varieties of cereals, vegetables and fruits have been developed by selection. So it has found following two types. First is your natural selection and second is your artificial selection, okay. So artificial selection is mainly of three types, okay, that is mass selection, pure line selection and clonal selection. So what is mass selection? Mass selection means basically a selection of a plant, and pure line selection, this is called artificial selection, okay. Okay, and the clonal selection means by the basis of cell, okay, we will select the cell of the variety, okay. So next is your multiplication and distribution. So as you all as I already told you what is multiplication and distribution, we have to multiply that species, है ना, the number of varieties which we having the extract product, and we will distribute according on the basis of distribution in the farmer, okay. We will distribute according to the quantity, okay. Okay, so next is tissue culture. So what is tissue culture? Plant tissue culture is improved and is the important field of biotechnology. The term tissue culture is commonly used in very wide sense to include the vitro culture of plant tissue, plant cells also, है ना, and plant organs also. But in strict sense, tissue culture denotes in vitro cultivation of plant cells and is utilized mass, that is callus culture. So in short we can say tissue culture is an experimental process through which a mass of cell, callus means an unorganized tissue, okay, is produced from an explant tissue, and the callus produced in this way we can use directly either to regrade plantlets to the extract of some primary or secondary metabolism, okay. This is known as tissue culture, okay. So I will also show you the example how, okay. So first of all we will go through the aims. What are the aims of plant tissue culture? So there are many aims, like to develop new plant from the plant organ other than seed, to produce hybrid variety of plant, to produce disease free plant from diseased plant, and to produce the period of reproductive cycle. So there are many types of many aims of plant tissue culture, okay. So what are the basic requirements of plant tissue culture? So first of all we will go through the laboratory, right? So for tissue culture or micro propagation, 3 such clean rooms are required where possibilities of contaminations are least. In general, space for the following is needed. So what we need, like washing, drying, storage, vessels, है ना, all these things we will need. The observation of the culture, how we will maintain the culture, है ना, that aspectic handlings of the explant of the culture, how we will do the preparation, the sterilization, the storage of the media. So all these things will come under the laboratory session. Okay, but so next is our apparatus requirement. So we need different types of apparatus like forceps, है ना, you can see here. So forceps, like needles, microscope, hot air oven, है ना, all these types of things we need for the apparatus requirement. Now lastly is your nutrient medium. Nutrient medium means we will select the pH or we will select the temperature, na, and also we will select the major types of elements, vitamins, and the sources or the main components for the tissue culture technique. Okay, yes. So here there are different types of cultures that they are all already present, like in which way what medium should be put, and in what quantity which solution can be take as a medium. Okay, so we will take the medium according to the chart. Okay, now next is our aspect condition. Okay, so what is the aspectic condition? So to avoid culture medium from the contamination of microorganisms, it is necessary that a complete aspectic condition is maintained, and around the culture equipment to the microorganism fail to contaminate the nutrient medium, the following sustains will be need. Like name of, first of all we have to make a surface sterilization. So for that we need name of the designifications, what the percentages we are taking, the duration of the treatment, means how what is the duration, in which duration we are doing which type of treatment. So all these things according to the chart. Okay, so we'll have to take we'll also take care of the light, right? Means in which numbers and whatever the maintenance for that medium, है ना. We also need the temperature arrangement is very necessary, है ना. And next is aeration means air mixed or dissolved in liquid, means how we can make that aeration, means whatever the liquid we are making, so that is totally mixed with that liquid or not. So all these things are required, है ना. So technique of plant tissue culture. So what are the technique? First of all we have to select the plant. Okay, then second is preparation of culture medium, means whatever the medium is required we will do. Or next is your initiation of the culture, means according to the light and the temperature we will put the culture. And then multiplication or subculture stages means we required, means whatever time, for how much time we are taking, for 2 weeks, 3 weeks, whatever the time is. And next is development of differentiation of subculture, means how it get developed, so you can see here, I'll show you, so this is the good example of representing of stages of the plant tissue culture. Okay, so you can see here, students, this is the cells of tissue organ. You can see this is the cells of tissue of the organ. Okay, we have to start this. Now what we will put, this tissue we have to put in the test tube, and that is known as explant, है ना. And we have already put it the medium. Okay, and we will plug the cotton on the test tube. Okay, so after some time what we will do, what we will see that this in that explant we will see a callus, है ना, after some time, and the culture tube, this is known as the culture tube. Okay, now subculturing, after subculturing it, what we will see, there is a part of the callus. See here, this is the small part of the callus, and after the organogenesis or the manipulating oxygen and the cytonin ratios, we will increase that ratio. what we will see, the initiation of the plantlet means a small leaf, small leaf, है ना. And after some day what we will see here, a new plantlet is growth inside that test tube. And at last what we will do, the potting, है ना, we will regenerate, we will put it in a pot, right. And that plant will become in a allied plant portion. And again we will do this technique once more. Okay, so this is basically known as your representation of stage of the plant tissue culture. Okay, so there are from the test tube to the field of test tube also, है ना. So there are types of tissue culture, there are many types of tissue culture like single cell culture, callus culture, है ना, protoplast culture. So there are many types of culture. So let us start with single cell culture. Okay, so what is single cell culture? Just look at this diagram. Okay, so in this single cell culture what we have to do is, the culture of a single cell on suitable sterilized culture medium means that, so we are talking about the medium means the solution, है ना, under controlled, and the aseptic condition is called the single cell culture. Okay, so establishment of single cell culture provide an excellent opportunity to investigate the properties and potentials of the plant cells, right. Several workers have successfully isolated single cell division and even raised complete plant from single cell culture. So there are historical events also we having, but we will go through the paper raft technique of single cell culture. So what we have to do, we have to take a isolated cell in the medium, and again we will put the cotton to plug that test tube, है ना. And what we will see here, we will put the filter paper raft, है ना, and this is called the cold callus. Okay, so after some time what we will see, the isolate cell on the paper raft, we can see here, and that old callus, है ना. And after some time again putting for the long time, what we will see, from that old callus we will found the new callus inside that medium. Okay, so this is the technique of single cell culture. So here some significance of single cell cultures are, single cell clones can be developed by this technique. We can develop a complete plant, can be developed by single cell culture also, means by this process also we can develop a complete plant. It is very useful for the development of improved varieties of the plant, means we can take any type of the variety of the plant. Okay, so single cell culture technique is most suitable for the metabolism studies, okay, or the metabolic study. And mutation plant can be produced by the mutant clones. So this is known as the single cell culture, this technique. Okay, so in our next class we will study with the callus culture. Okay, thank you.